Hello, everyone. My name is Ali Reza Hagigi. I'm the founding director of International Center for Genetic Disease and an assistant professor of medicine at Brigham and Women's Hospital and Harvard Medical School. It's my great pleasure to welcome you to the second lecture of the Harvard Keynote series on genomic medicine. Today, we are delighted to host Professor Sir Mark Caulfield, who has made substantial contributions to the discovery of genes related to cardiovascular health, cancer, and rare diseases. He is amongst the top 1% of the most highly cited researchers worldwide. And today, we have participants from 29 countries. This series is organized by the new International Center for Genetic Disease, or ICGD. ICGD focuses on the analysis of subjects from different parts of the world for research into the genetic causes, consequences, prevention, and treatment of disease. ICGD has been formed by Brigham and Women's Hospital and Harvard Medical School in partnership with other Harvard-affiliated institutions, including Mass General Hospital, Boston Children's Hospital, Harvard School of Dental Medicine, and the Broad Institute. We are delighted to have a distinguished board from all these institutions. I would like to thank the leadership of the Brigham and Women's Hospital, Department of Medicine, and Division of Genetics for their continued support. I'm also thankful to our scientific and admin teams. In this series, we feature talks by internationally renowned scientists as they explore the very foundations of life and improve our understanding of genetics and biology. This series is currently scheduled for the third Tuesday of each month from 9 to 10 a.m. Eastern Time, and it will be on Zoom. Today, we are delighted to host Sir Mark Caulfield, who is Professor of Clinical Pharmacology and Vice Principal for Health for Queen Mary's Faculty of Medicine and Dentistry, and the CEO of Bart's Life Sciences. Mark Jonathan Caulfield was born in 1960. He graduated in medicine in 1984 from the London Hospital Medical College and trained in clinical pharmacology at St. Bartholomew's Hospital, where he developed a research program in molecular genetics and hypertension and translational research. Over his 40-year career, he has made substantial contributions to the discovery of more than 2,000 gene regions related to blood pressure, cardiovascular health, cancer, and rare diseases, for which he has been awarded multiple prizes. His research has changed national and international guidance for high blood pressure. Dr. Caulfield was director of Queen Mary's William Harvey Research Institute between 2002 and 2020, and was elected to the Academy of Medical Sciences in 2008. In 2013, Sir Mark was appointed chief scientist for the Genomics England and delivered the 100,000 Genomes Project using whole genome sequencing in rare disease, cancer, and infection, from which he co-created the National Genomic Test Directory and a new genomic medicine service in the UK. This offers equitable access to genomic testing across 56 million people, and 500,000 whole genomes will be sequenced in direct healthcare over the next 5 years. In 2019, Sir Mark was awarded a knighthood for his leadership of the 100,000 Genomes Project. In July 2021, Sir Mark stepped down from his role as chief scientist for Genomics England to lead Bart Life Sciences as a partnership between Queen Mary University of London and Bart's Health NHS Trust. This is driving the development of a world-leading life sciences campus at Whitechapel, focusing on digital precision healthcare. He is president-elect of the British Pharmacological Society. Sir Mark's lecture will be 30 minutes, and then we will open the session to audience to ask questions. It's a great honor to introduce Sir Mark Caulfield to you as today's keynote speaker in our ICGD Genomic Medicine Lecture Series.
Thank you so much, Ali Reza. I'm just going to share my screen. It's a pleasure to see everybody online from so many different countries. And hopefully one day shortly, we'll be able to get together as we used to do and enjoy these meetings as people who are scientists exchanging knowledge. I'd like to thank colleagues across the Harvard family and Boston for all their help over the years, both with cardiovascular research, but also their collaboration and friendship, particularly during the 100,000 Genomes Project. I have been largely a government public employee for the last 37 years, and therefore I don't really have any disclosures, although I am advising the United Arab Emirates on their national program, and I do advise a number of programs around the world, but that's for free. So in the United Kingdom, the government has a strategy called Genome UK, Our Future Health. And what you can see here is that on the left-hand side, we have what we're doing in diagnosis and personalized medicine and direct healthcare. That's the 100,000 Genomes Project and the 500,000 whole genomes Ali Reza referred to that will be sequenced by 2024. We have a new cohort that's starting in the middle in the green. That's now called Our Future Health, and that's 5 million participants. And that will start pilots in the summer. And we also have the 500,000 people from UK Biobank, which many of you know well and are working on, and there are now 200,000 whole genomes available. We also have an NIHR bioresource, which is 200,000 people with different diseases who are deeply characterized. And that's our national genomic strategy. And we want to use the research on the right to drive into the healthcare on the left. So the 100,000 Genomes Project arose from the London 2012 Olympics. It's an unusual Olympic legacy program. There were no grants written about this. And the task set by the British Prime Minister then, David Cameron, was to sequence 100,000 whole genomes from direct healthcare. And it needed to be tractable to benefit in the health system. So we worked on rare disease, cancer, and infection. And that reached over 97,000 people. That's 40 petabytes of data. A lot of what we had to do in the first 18 months or so, and this is where my colleagues here on this call was so helpful in their advice and the information they gave us freely, was to create a structure within our health system to do this. And at the peak, we had 13 genomic medicine centers. There are now 7 genomic medicine service alliances. So we've condensed them to 7 hubs. And about 5,000 frontline NHS staff worked on this program over the course of their working week. And we openly advertised in Nature, and a coalition of over 3,000 researchers from 33 countries have volunteered to help us drive up the value of these data for clinical care. I'm going to focus in on rare inherited disease. And we published in November 2021 in the New England Journal of Medicine our preliminary findings from an early pilot study that we did on 2,183 families. They covered 161 disorders. In the main program, we worked on over 1,200 disorders, and there are 36,000 families in the main program. And what I first realized was that we needed to standardize eligibility and clinical characterization. So we anchored that, as you might expect, in the human phenotyping ontology. So my data can speak to your data because each of us may only have 1 or 2 families with a disorder. And if we are able to put that data together through federation or whatever tools like Matchmaker Exchange, we will be able to solve those cases. So we semi-automated the analytics, and we got the NHS involved. And initially we were using gene panels. Now we range more widely across the genome, and the NHS closed the cases, so they have to tell us what they did with the information. This is one of the children who got a benefit through the program. This young girl had GAMT deficiency. She had no genomic diagnosis. She presented with intractable seizures and intellectual deterioration. And she was so locked in, her mother was convinced that she was going to be in a wheelchair and then subsequently almost certainly die. Simple discovery of a mutation in the whole genome allowed this child to have a new diet with creatine, ornithine, and arginine. And the child is now a completely different person, has no seizures, and has had much intellectual improvement. What if we could have discovered this earlier in life? At the very bottom, you can see the different numbers of families we have in the different systems. And what that means is we have a large critical mass available and open to you from anywhere in a bona fide institution worldwide to do research on. So let me just give you another diagnosis from the program, and you can see this child's video on our website there. I'll send Ali Reza the slides and he can distribute them if he wishes to. This is a 4-year-old, and this is an example of where a whole genome changes a clinical diagnosis. So this boy had presented to West London with anemia, developmental delay, and short stature, and had been given a diagnosis of diamond black fan anemia. But because of his intellectual disability, we ran a panel that looked for causes of that, and we found a de novo mutation in thyroid hormone receptor alpha. That's a treatable form of thyroid hormone resistance, where the receptor is resistant to normal thyroid levels, but you can overcome that with dose titration. So he now is on a dose titrated regime, and if you look at the video, you'll be able to see the evolution of this boy across his treatment, and by the end of this, having been only able to walk outside with his parents' support, he's dancing. So what if, again, we could have found this earlier? And this child, as is often the case, and this audience probably doesn't need this to be said, but this child has enabled a further 7 families to be diagnosed. So just from the New England Journal paper, I wanted to highlight some things. 14% of the diagnoses that we made were outside of the exome and would not have been detected by exome sequencing. On the left, you can see the variants that we would detect in conventional panels or in an exome. In the right of the pie chart, you can see in yellow, green, and orange things that we're finding because they're in the whole genome. Across any disease in that pilot, we achieved about a 25% diagnostic yield on average. Some of those disorders were very challenging, and there are areas where we've never made any progress to date. Here you can see the impact of a singleton, a duo, that's usually a mother and a child, a trio, and so on. And you can see that the larger the family structure, as is well known, the better the chance of a line of sight and an answer. Interestingly, what we established here and have continued is a research ecosystem alongside direct healthcare. And what that's done is in monogenic type diseases, it's driven up the discoveries up to over 35% as a result of additional research. And also in disorders which have a more complex or oligogenic architecture, we're at about 11%. This is the impact of Exomizer, which integrates multi-phenotypic information from various models and humans. And what this has led to is an improved precision of prediction of deleterious variants. And this is in the paper, but is now a routine part of our pipeline at Genomics England. And we use this on every family we look at. Here is the diagnostic yield by family, sorry, something's lost there, that's it. So what you can see here is that although we have an average diagnostic yield of 25%, you have many diseases, hearing, metabolic, intellectual disability, neurological, or ophthalmological, where the diagnostic yield is well above that. And in some traits, it's up at 60%. And on the next slide, what you see is we looked at these, the case notes of these children and adults, and there are a lot of adults in this program at that stage. And whatever you did, whether there was prior testing or no testing, you got about a 30% diagnostic uplift. And here's the performance of chromosomal tests versus whole genomes. This is the uplift from a whole genome. Targeted tests, NGS panels, and a small number of whole exomes. But whatever the test, a whole genome was better. What is the diagnostic odyssey of these children? So these families, if they were a child born after 2003 and enrolled in the project, they spent 68 hospital appointments in 6 years without a diagnosis from usual care. Their unaffected relatives, over a much longer period of 120 months, had only a median of 18 appointments. Post-diagnosis, what you then see is the appointments become fewer, much more focused, because we have a clue to what the diagnosis is, and that can reprioritize therapy. They used 183,000 episodes in total of hospital care via various aspects of the hospital system. And they cost 87 million, that's £15,000 per participant. So of course, a genome at a few $100s doesn't look so expensive. And because this is buried in the health system, we often don't see this true cost. Of course, there are other siblings unaffected had also healthcare contact. So they had some contact, but it's at a much lower rate. And this doesn't include visits to the family physician or disease treatment costs. So what did we do with this? 25% of the diagnoses had an immediate clinical utility according to the National Health Service. And you can see that for some it changed medicines, it led to additional surveillance, some were eligible for trials, some could inform reproductive choice in the future, and 32 got a dietary shift or vitamins. Importantly, there were 829 genome-wide significant signals and burden testing. And here is, in my view, the battleground. And we think there are 579 potential diagnoses, but we do not have sufficient evidence to return those as definitive diagnoses. Though with research, 22 have been converted either by us or others to clear-cut diagnoses for some of these patients. And we continue to generate that from research endeavor every day. So the totality of this pilot cost the NHS over the lifetime of the individuals £106 million. So all of a sudden, a whole genome doesn't look too expensive. That's $138 million. We also published about 3 weeks ago in Lancet Neurology our work on Expansion Hunter. Expansion Hunter measures repeat sizes. And so what you're seeing on the left is a histogram of the normal repeat size in the population in general. And then to the right of the serrated line, the triangles are repeat expansions. And they have different ataxin-based disorders, or Huntington's is the bottom row. Let's focus in on Huntington's. The child out near 100 repeat expansions, which we didn't think we'd be able to measure with short-range sequencing. What we did here was to validate our whole genome sequencing against known positive cases, and then we evaluated it in the 100,000 Genomes Project. So this child is under 10 years of age and has Huntington's disease, but that was not diagnosed in the health system because it was never suspected. His father is over here on the left, and he's around just above 40. So we diagnosed the child and realized the father had the repeat expansion at exactly the moment the father got his first symptoms. So this dramatic repeat expansion, it's well known, has occasioned a much earlier presentation. And so the genome is the ability to find things that we don't suspect clinically. When we go to the main program, this is 36,000 families, we're at about a 20% diagnostic yield. And the reason for that is the research endeavor is still a work in progress, and that is yielding new diagnoses every day. I'll move to cancer briefly and just share with you, there are 17,339 cancer participants, and these are all fresh tissue whole genomes and germline genomes on all of these people. It's the largest single publicly available cancer resource for researchers worldwide, and I encourage you to come and work on it. I just want to highlight you a case here. So this is a 42-year-old who had HIV and previous hepatitis B and presented with a very large dense lesion in his liver, which turned out to be colorectal cancer. After usual care testing, no findings were made that would inform his clinical care, but he received usual care chemotherapy and the lesion just expanded and he became symptomatic. So we did a whole genome, and this turned out to be a mismatch repair defect, and this person had Lynch syndrome that had not been noted or presented. What if we could have detected this earlier? And he got to have a trial of immunotherapy as a result, which he would not have received from normal care. So that's an example at individual level. Here's an example of cancer of unknown primary. And what we found in this person's brain metastasis was that they had various mutations that might be associated with a non-small cell lung cancer, and further looking at the tumor mutational burden was compatible with lung cancer. And then we further looked at the signatures, and there was a benzopyrene smoker signature. And so a genome can help you to get from a cancer of unknown primary to a primary diagnosis. So this is what the NHS did with the results we sent back in cancer. So about 25% of those results went back to a genomic tumor board. And you can see on the right-hand side that some of them had DPYD and therefore didn't get or had reduced dose of 5-fluorouracilocapsidine. Some had licensed and unlicensed use of drugs that they wouldn't have got through usual care. 204 got into a trial, 43 had high tumor mutational burden, and if they recur, are prioritized for immuno-oncology, and 69 were referred back to clinical genetics. So this is usable in the clinic, and therefore of value. Here's something we've been doing more recently. We've been using routinely, and we life course follow all of these people with every registry, every piece of health data we can find. And this is a pathology report that we get from one of the registries. And so we machine learning text mine this to extract the information we need to stage the patient and to describe the tumor in clinical sense. And I'll show you what we've done with this data. So this is replication from real world data of a clinical trial signature reported in 2012 in breast cancer. And this is PIK3CA wild type here at the bottom in pink with a worse prognosis, and the mutant with a better prognosis. And this is from real world data. So we can recapitulate clinical trials just from routinely collected health data and chart the prognosis. And we do this now for many people. We have a cancer paper and advanced presentation, which also observes some favorable signatures where there are about 20 or 30 patients who suddenly do very badly. In pharmacogenomics, we've looked at over 76,000 whole genomes, and 99.5% of the participants have at least one gene-drug pair, where if they come across the medicine, it will either be inefficacious or harmful. And 25% of the participants have at least 4 of these haplotypes. What we're doing now is to go on within known pharmacogenes to look for rare variants which might alter the phenotype. And given that 6.5% of admissions to United Kingdom hospitals are due to adverse reactions, and a proportion of those will be due to pharmacogenetics, what if we could wander around with a pharmacogenetic traffic light system on a mobile phone, type in the drug, and understand which we should or shouldn't receive? Now, we've assembled this coalition of intellects, and you're all very welcome. In blue is where they come from. In grey is the work in progress, and I'd love to see folk from those areas coming in to work on the data. But there are over 3,600. There's about 500 working on a regular basis in the data center, and we're moving that gradually to a cloud-based system so it'll be easier to federate. That's 33 countries, 413 academic institutions. They have won themselves 50 million grants for their own research, and we also work with companies. The really great thing about this is over 300 diagnoses have been made by these people. So this ecosystem is fueling return of results to the clinic. So what do we do with all of this? Well, in the UK, there's good evidence that it can take between 9 and 16 years to convert a clinical trial into actual use in the clinic in the health system. We couldn't do that. We had to leave a legacy that had not meant that we'd marched people to the top of the hill with whole genomes and then left them there. So in 2016-17, we began planning a new genomic medicine service, which we launched in 2018-19. And that is 7 genomic lab hubs, a national test directory where we reprioritized 300,000 tests, upgraded 25% to new technologies, and 500,000 whole genomes are available for direct NHS care, and Genomics England provides the sequencing. Every patient in this service is offered genomic consent for research. That's for life course follow-up, recall for research, and access in a secure data center based in the UK to international researchers and industry so that we can drive the value up for clinical care. So last week we published in Nature 7,491 whole genomes from severely ill COVID-19 patients from 224 UK intensive care units, compared to 48,400 controls. We detected and validated 23 signals. 16 of those are novel. So those of us who work on complex traits will know this level of discovery from such a small number of people is unusual, and some of these have bigger effect sizes than normal. Importantly, in multi-ancestry analysis, we found 3 signals that were driven by all ancestries, but particularly driven by South Indian Asians, although on their own, they don't pass genome-wide significance, and they're TRIM46, BCL1A, and LINCO1276. The genes are seen here in the Manhattan plot at the top. This is the multi-ancestry Manhattan just down, highlighting the genes I've just mentioned. One I'd like to draw to your attention, because this is a rarer variant, which we couldn't easily replicate on the chip, but we did manage to replicate using exomes from Regeneron, which we're very grateful to. And this was validated in the Host Genetics Initiative. And we've calibrated the p-value for the number of independent tests, which is 2.26 million. So we've adjusted the p-value. So here you see these genes. What you'll find, there's also factor 8, which isn't mentioned there, but you find these genes categorized into 3: inflammation, immunity, clotting, and blood groups, which I put together. So that may explain the coagulation effect of COVID. We've also brought live a rapid exome service for children in the NICU or PICU, and that has a 37% diagnostic rate in the NHS. We are working on bringing whole genomes live, but that's a work in progress, and the time delay is the important thing here. So in 2018, I chaired a group. Some of you were represented, Robert Green, others from the United States helped us formulate advice to the Chief Medical Officer that it could be right to examine by research whether a whole genome has a role in improving diagnostic yield in early life, and perhaps diagnostic yield where there could be treatments. So we thought about this as that the genome is the raw sequence, the assay. The analysis is what you do, and you can choose to do different things at different points of somebody's life. And the return of results is the output. And again, the output may be different at different times in someone's life. We focused the program on treatable genes, and with David Bick, David Dimmock, Richard Scott, and Tom Fowler, we developed RX Genes. David Bick really led this. You can download the app from the App Store. Do tell us if you find any errors in it. But this is an app with treatable genes. So genes that there is a treatment for that's been identified. So this is what you do. You plug in phenylketonuria and it comes up with low protein diet. It's all pretty intuitive stuff, but it's a useful single source of the truth. And there wasn't one. So we looked across rare disease, and we've identified about 639 disorders where we can identify about a 1 in 190 frequency of births. That would be 10 children born every day in the United Kingdom, or 54 children born every day in the United States. And these children have a fully penetrant disorder that it will present before the 5th birthday, and there is a potential treatment. And only 8% of these are expensive. We've also looked at the economics of this. And because children have so much life left, you can gain a dramatic increase in quality adjusted life years gain, bearing in mind that about a third of children with a rare disease will die before their 5th birthday. And so you are approaching, but not quite at cost effectiveness, and further work's needed to be done there. But I can tell you that the work that I did leading up to this has been funded now by the United Kingdom government. And we've just been out and publicly consulted, which we published in June 2021. And this shows on the left what the public and patients thought about this. They were excited. They wanted it to happen. They wanted it to be equitable and involve a representative sample, and to be transparent, and to really test whether whole genomes could improve upon the 9 measurements we make from our heel prick test that we do within 1 week. So Genomics England will continue to work on diversity in genomics, cancer, and has a new program in newborns, which we're starting pilots on immediately. And we'll continue to work on rare disease and provide whole genomes. So, ladies and gentlemen, the National Health Service is creating a national genomic medicine service based on this, providing consistent and equitable care for 56 million people, standardized with specifications, protocols, and a single standardized genomic consent. And this is delivering against an approved national test directory for which they're reimbursed, covering the use of single gene to whole genomes in the NHS, and building a single UK-based knowledge base. But this knowledge base is available to all of you across the world. And I do encourage you to come and work on it, because I firmly believe that the future is a global coalition of intellects. And that's why I'm delighted to be working with colleagues here from Harvard and other universities across the world in the Global Alliance for Genomics Health. I should thank Ali Reza and you all for listening to me today. These are the most important people to me. They're the participants. I would be delighted if you come and help me find the diagnoses that we haven't yet found for them. These are the team that wrote the New England Journal paper. I should pay tribute to Damien Smedley, who co-led it with me. And these are our funders. These are the team that wrote the Nature paper. And I should pay tribute to Thanos Koutsanis at Genomics England and Kenny Bailey from the University of Edinburgh. And I thank you so much for your attention. Please do email me if you don't get to ask your question. Thanks so much, Ali Reza, for having me here.
Thank you so much, Sir Mark. That was an amazing presentation, and thank you for accepting our invitation to present at our keynote series. So now we open the session to attendees to ask questions. If you have a question, please raise your hand and you will be unmuted and you can ask questions. And please feel free to tweet about the talk and the questions as well.
Hello, Dr. Mark Caulfield.
I would be thankful if you introduce yourself in just your name and your affiliation and ask questions. Thank you.
Okay, sorry. My name is Dr. Balwe Mathru-Ball. I was formerly at Nottingham University Hospital and part of the 100,000 Genome Project, but I'm currently now the director of the Biobank in Abu Dhabi. So, Dr. Mark Caulfield, so lovely to see you again, and thank you for a wonderful presentation. May I please ask, because Abu Dhabi is considering rare disease genomics planning as well, how important is it for you, do you think, that we actually recruited the trios into the rare disease program, if you can?
So Balwe, first of all, thank you so much for your wonderful work in Nottingham. We couldn't have done it without you.
Thank you very much.
In the advice I've been giving, so I'm advising a group who are advising the UAE government about the program, and I've advised them strongly to focus on things that will be tractable to health gain. I think it's particularly important in that area of the world where there is greater closely related intermarriage, that we actually do a really fantastic job for people with rare disease. Partly that is because I think that we need to accelerate treatments and treatment opportunities for people with rare disease, and having the platform of things like the 100,000 Genomes Project, the UAE Genomes Project, in the United States, the Undiagnosed Diseases Network. These are vital platforms to allow us to find the cases that can actually participate in the studies, and also that we can actually shorten the patient funnel to enroll people in studies and make these therapies more affordable, which is vital. So Balwe, I've strongly encouraged a focus on things that are tractable to direct healthcare in UAE, but obviously they are also looking to sequence the whole population there. And there's others here probably on this call who've been involved in that as well.
So I'm part of the Emirati Genome Project as well.
I guess that, fabulous. Well, you'll be a fantastic help to them.
Thank you very much again. Thank you for answering that question. Really appreciate it.
Thank you.
Hi, I'm Gabriela Repetto. I'm a clinical geneticist at Universidad del Desarrollo in Santiago, Chile. We've coincided through G2MC and others. Thank you for the great talk. Very jealous of what you have achieved. So my question has to do on what are your thoughts for low and middle income countries or countries with few genomic resources, and how could they leapfrog what strategies are there to leapfrog the introduction of genomics into healthcare, and what have you seen in your advisory roles for different countries around the world? Thank you.
So I think Gabriela, this is quite important, and I think this does require those of us who are in more affluent countries to reach out, not only offer partnership, but I actually, dare I say, also offer money, because it's not going to happen otherwise. One, I think of the initiatives that has been transformative due to investment, but also has been H3 Africa, in binding the African continent together. I'd be very keen to see something else like that in South America. And if you think I could ever help with that, I'd be really happy to do so. The UK is not the biggest investor in healthcare, but what I think is one thing that I would try to do is to capture the imagination of politicians, because although we may be somewhat cynical about our politicians, the practical reality is when they actually suddenly decide that something's really, really important, they can put the weight of government behind it, which is what happened here, and present cash in a disruptive manner. Now, if business as usual had occurred in the United Kingdom, we would never have done a whole genome, not even by now. So we need disruptive initiatives. So I think one thing is to get the excitement of government, but I think the other thing is to turn up with somebody else who will support you with funds. I don't have immediate ideas, but I'd be happy to have a conversation with you about how we could help. I firmly believe that this will only be done properly when we've democratized access to the entirety of humanity of genomics, where it's needed and where it's got a clear evidence base in healthcare. And that's why I like the work of George Church in trying to bring the Personalized Genome Project to people and open the doors to opportunity for folk who may never have considered this.
Thank you. So next question. Dr. Jihad Al-Kathali.
Yes, this is Jihad Al-Ghazali from Abu Dhabi, from United Arab Emirates. Thank you very much for your great talk. Really, we need to invest more and increase the awareness of the medical community about precision medicine or individualizing our approach to therapy in patients with different diseases. And no doubt that genomic medicine and advances in genome medicine has led to or has denied the concept that we used to adopt in the past, that one disease phenotype or can be treated the same. But now with precision medicine, our approach is becoming completely different, and we are individualizing our therapy to different individuals. There is one point that I want to raise about your approach, the whole genome sequencing and whole exome sequencing that you have been using for diagnosing many of those diseases. Have you considered incorporating CNVs, copy number variants, as part of the whole genome sequencing and whole exome sequencing? Because this is really very important. If we can incorporate the copy number variants together in those tests and evolve our whole genome sequencing and whole exome sequencing, this will contribute a lot to diagnosing a considerable percentage of diseases that we usually miss with whole genome sequencing and whole exome sequencing.
So Gerd, thanks for your question. We look at all variants, including copy number variants. I didn't show you today, but we have many diagnoses that we've made with CNVs, and some of these would not have been seen by other orthogonal tests that we routinely use. So you're absolutely spot on. Also structural variants. Now, long reads, I think, will improve the fidelity of this. So we have some data working with Oxford Nanopore that pore-based technologies can give us much greater structural variation than we suspect from short read technologies. So I think there will be certainly a place for long read technologies I think it's coming, it's almost upon us. To your earlier point about mainstreaming this is the only way it succeeds. We have to open this up. So alongside the program I was describing, we invested £25 million in educational programs, and over 1,000 people have been through masters in genomic medicine. But you have to mainstream this. The other thing we have to do is to improve decision support, because there are too many people who go through multiple contacts before anyone thinks of a rare disease. So I have a child in the program, which I've looked at the case several times. I didn't show it today, but they went to the hospital 25 times before anyone did a genomic test. And actually, when you showed that to a room full of people who were geneticists, they said, why didn't they do the test on visit one? And it's because people just didn't know what they were looking at. So you can't make this work if you don't mainstream it and you don't support decisions in the health system. And that's vital.
I fully agree with you. And this is, in fact, it is reflected in our present practice, where you, especially when you are responsible for analyzing data, raw data. Yes. And the importance of artificial intelligence, like teaching your machine to incorporate the phenotype of the disease, and like other measures like inheritance patterns, and all those, they will help, in fact, in the diagnosis and application of artificial intelligence. It is coming to our health system, and it will be part of our daily practice, but it will take some time.
I completely agree, and also go ahead. At the moment, we're using multiple modes to analyze these data. So expansion onto for repeat expansions, other CNV and structural variant measurement programs, and then looking, we've been using Splice AI. So we have several diagnoses from Splice AI, where you're comparing essentially the intron exon boundary of everyone in your data asset with the person with the disease. And we found several intron exon diagnoses as a result of that. So these technologies, we will have multiple analytic pathways. What we do in Genomics England, we put that into an accredited, like CAP type accreditation, or ISO if you use that. So we have an ISO accredited pipeline. So we get the evidence that they can be used in health system, so we can return that to the clinic. But thank you for your questions.
Thank you. Adrian?
Thanks so much. That was an incredible talk, Dr. Caulfield. I'm curious in regards to structural variation specifically. You mentioned again the potential use of things like nanopore-based sequencing in an effort to capture those more effectively. So I'm curious in terms of the current paradigm, is your team currently evaluating structural variation across the genome using whole genome sequencing? And in regards to the future, do you believe that there's an ability to sort of improve training of the detection through sort of algorithmic means for whole genome sequencing, or does this require sort of multimodal nature of sort of integrating long read approaches with a short read to more effectively capture the structural landscape of the genome?
Yes, Adrian, great questions. Just to answer the first two, we, for about over a year now, I've left Genomics England, so I'm not in charge of the program anymore, but during the early phase of the pandemic, we started a major program with Oxford Nanopore in both cancer, and we're now doing work in rare disease. And what the team see is that you with long read technologies, you do discover much more structural variation than you would have expected. Those technologies are a work in progress. We're also working with Pacific Biosciences, which some of you will have seen with the genome in bottle work. They've got very good long range, even coverage across the genome now, and that technology is maturing. And there will be other pore-based technologies that will come to life. And although we have worked principally with Illumina in a partnership, that was because Illumina were ready to do this. But we do see the possibility of several states. One is either there will be an intermediary state where we use long reads alongside short read technologies, or that eventually we do go to long read technologies. And we can tell that the commercial entities think that's important because of the IPO of Oxford Nanopore, which was highly successful, and the fact that Illumina are talking about longer lengths of reads. So I think all the commercial entities perceive this as important, as we do as a scientific community. It's still a work in progress to get those technologies to clinical grade, if I'm honest. Illumina short read is clinical grade. At the moment, we don't have Nanopore as used as clinical grade, but I'm sure the Nanopore and PacBio will follow. The other thing that will also present to you and I, as people focusing on that, is a competitive tension in the market. And I think that's pretty healthy for innovation. I also think it's incredibly important to get it in the health system, because some people still believe this is too expensive. But I think multimodal algorithms are the way forward. And I do think that we'll be using a lot more advanced analytics in our interpretation of a whole genome. The other thing, Adrian, I also think is the battleground now is we can create millions of variants really quickly now. The genome sequencing is really the battleground. The battleground is what's the function. And that requires a coordinated effort. And you've got one in the United States. And I hope there'll be one here in the UK, because unless we convert these variants of uncertain significance into functionally characterized variants that are usable in the clinic, we're doing our patients a disservice.
Thank you, Adrian, and thank you, Sir Mark. Dr. Morton?
Hi, thank you so much for this inspiring talk. And I just wanted to say that you know how happy I am for the use of the genomes in newborns. And I just wanted to make one point in terms of whether the disorders are fully penetrant or not. We recognize now that some of the sort of highly regarded variants that occur in hearing, loss, such as GJB2, aren't always penetrant at birth. So those kids are missed, and every day we're missing them here. And I think those are kids who would totally benefit from having their newborn genome done in terms of picking up hearing loss. So look forward to that. Thanks.
So Cynthia, it's first of all lovely to hear your voice, and thank you for all your personal help over the years. You're absolutely spot on. So to kick off the program, we are focusing on fully penetrant conditions, but I'm aware of some variably penetrant conditions where the professionals like you who face these patients may say, actually, I think you should include this, and you should include this because if we don't find it, a child may spend time at school not being able to hear in the class and nobody notices it. And I remember very well you telling me at one meeting, I think it was in San Diego, of American meeting of human genetics, that that some of these kids even passed the hearing tests initially, and then their hearing deteriorates, and they cannot cope in an open classroom. So we're about to do the professional consultation on what should be included, and I think people will make the argument for variable penetrant conditions as professionals, and if the profession make that argument, I think that's a good basis for going forward. So we haven't ruled out variably penetrant. And actually, our friend later, Hudson Alpha, David Bick, is now the director of the newborn program. So our newborn program is led by an American.
Thanks.
Thank you, Cynthia. Dr. Riyad Bayoumi.
Thank you very much. My name is Riyad Bayoumi. I work at Mohammed bin Rashid University in Dubai, UAE. My question is, are you going to extend the work towards the common diseases like diabetes and hypertension? Because, yeah, I mean, a lot of research is going on, but a database of that magnitude would make life very easy for researchers working on common disease, schizophrenia, diabetes, hypertension, and so on.
So Riyad, this is a very, very good question and very timely. So Biobank UK have released the first 200,000 whole genomes. All of us is busy sequencing their 1 million cohort. This is going to become a reality and available. What I can say is from our COVID work that we did, you might say, you know, why did we do whole genomes? Well, the bit that we haven't yet done properly with the COVID work is proper interrogation of rarer variants. And what we did see is, and this will be true for complex traits, is that because you have a much more granular picture of the variation across the genome, you can fine map with a whole genome. So there are at least 2 peaks of those 16 loci, 2 out of the 16, that only exist because we used whole genomes. We would not have found them as separate loci. We wouldn't have been able to separate them with a genotyping chip. And we've also done, as TopMed has, produced an imputation scaffold for, in this case, UK Biobank. And we've just returned UK Biobank data with a much more granular imputation to Biobank for issue to the community worldwide. And I can tell you, just in my own field of blood pressure, we ran blood pressure traits. It's the 500,000, you know, the world and its mothers worked on that data. But with advanced imputation based on whole genomes, you can discover more from that same old genotyping chip with the 1 million variants. So the bottom line is whole genomes are going to add, I think, value in common complex disorders. The other thing I don't think we should underestimate is the role of variants we have not characteristically typed in common complex disease. We should not underestimate those. I can see strong potential roles. Look, we've already got evidence in autism for roles of copy number variation. And I think that whole genomes will present us with an unprecedented opportunity to understand the role of both common and rare variants. The other thing I think that a whole genome brings to the population level, and I haven't got proof of this in the COVID work yet, but in fact, our next bit of the COVID study is to study 14,000 severely ill people. And we've now got phenotype, this was hard to do actually, 14,000 mildly affected or asymptomatic COVID positive patients. So what we've designed, I've been busy designing an extreme phenotype comparison, because there's some evidence if you maximize on extremes, you'll improve your power. And that's the next experiment we're going to do. And we will do proper gene burden testing, because in the first Nature paper, because we wanted to get the data out, the team said, well, we've done 6,000, we should stop. No, if you do gene burden testing, if you want to really find rare variants, you've got to have enough people to find the rare variants, or you're not going to find them. So we need to use the whole 48,400. Now, with resources like Biobank, with 200,000 whole genomes involved, you and I can continually doing that. So we can have our case-based resources like schizophrenia or whatever, as long as we're sensitive to population and ancestry in our analysis, we can actually do things that we wouldn't have imagined. And what we do need, and that's why your project in UAE is so important, is we need more nations around the world to represent the true richness of the diversity of our planet, because otherwise some people will be left out and disenfranchised from genomics and its role in precision healthcare. And I think in common complex disease, the big hope has got to be that we'll identify new therapeutic targets using that approach.
Thank you very much.
Thank you so much, Sir Mark. The next question is by Dr. Saeed.
Yeah, hi, thank you, Ali. Thank you for your talk. And to justify a whole genome sequence over exome sequence, how often and how far you are finding intronic variants away from the splicing junctions and relevant to the phenotype? That's one question. The second question, how often you had a chance to look at NGS report, whole exome sequence, from all of these commercial companies all over the world, and you felt this is useless?
So Saeed, yeah, an important observation there at the end. So let me go to your first question. The bottom line is, in the New England Journal paper, 14% of the diagnosis lie outside the coding region. So they're either intronic, 5', 3', or intergenic. And we did find the intergenic things. Now, when we did that work, we were not great, if I'm honest, at doing that. We're getting much better. And I think we will just find more and more things. To Adrian's earlier question, I think that does require us to be able to automate some of these analyses and use more artificial intelligence type work. But that requires us to build an appropriate database of genomes to work on, or we don't have the substrate to optimize those and refine them. To your question about commercial entities, I've limited experience of this, but I have seen the reports you're describing. One of the things we have to be careful of as a community, if we're facing healthcare and our patients, is first of all, let's do the best by the person in front of you. And I think that some of the reports leave a lot to be desired at the moment and could be better. But I do think that that will only come by standardization and the community saying, as we've done, for example, in GA4GH for certain analytics or certain types of pipelines, this is a standard. We all agree this is a standard. If you're not at that standard, you shouldn't be using this in healthcare. And I do think that we have to see those type of standards. And to return to the health system in the UK, if you're not ISO or CAP accredited, you will not be returning a result. They will, they have to treat it as research. And, so if it's really definitive, then they might use it. But if not, what they'll do is they'll take a look at the report and they'll repeat with an orthogonal test to validate or work out what's happening. And so, Saeed, you're right about standardization, and that is vital to the acceptance of this broadly. If we mess up on standardization and your genomes are apples and mine are pears, that's just going to set us back.
Thank you very much, Mark. I have a follow-up question on this. So now we do have more effective tools like Splice AI for detecting splicing variants. But we also know that Splice AI is more accurate. The more you, the closer you get to the splice, to exon-intron junction, is more accurate.
Yes.
So did you identify a lot of, or what percentage of the splicing variants you identified are like deep intronic, and did you validate them?
Yeah. So everything we've reported has had to be validated by an orthogonal test. Ali Reza, it won't surprise you to know amongst the 500 or so that I think are plausible diagnoses are many things in introns and splice junctions where I could not get the level of absolute evidence to allow me to return that to the health system. So in Spice AI, working with Illumina, we found about 40 or 45 events which we think are genuine causes of disease. You're right, it works better closer to the junction. In intronic, I don't have that in my head, but it's probably between 3 and 4% out of the 14% that were outside the coding region. So 3 or 4% of that were intronic. Now, what were these things? They were unexpected regulatory regions by and large, or they disrupted, they were at the intron exon boundary and they were slightly disruptive of a splice site. So it was read through, but that's really a splice junction anomaly or that's what we would as a community call a splice junction, but they were in the intron. You know, I think we're about to do a complete pan-rare analysis of all 36,000 because our report in New England was preliminary. So we have to do the final report, which is a further follow-up paper. That's work that's just starting, but we're combining a stronger ecosystem of using the advanced analytics suite of tools in this occasion. And also we're just completing 5,000 transcriptomes on undiagnosed cases because I do believe that transcriptomes will add value. And we've also been looking at high abundance and low abundance proteins by mass spec and using active-based analytics like Somalogics and Olink. But those data have just arrived and are just ready for analysis. So we're about to do that piece of work. I have no idea how that experiment will turn out. I would probably, I'm not a gambler, but I'd probably bet Alireza that we will find a few diagnoses clinched with transcriptomes as others have done. But we'll have to wait and see. And this time, Ali, we're going to spend a lot more effort on the non-coding region. So everything that isn't an exon, we'll be spending a lot more time on it because I don't think we've got the full value of the whole genomes yet. In fact, I'm sure we haven't.
Thank you so much, Mark. Dr. Afaf?
I think, Dr. Afaf, you're muted.
Yes, you are on mute.
Hi, thank you, Dr. Ali, and thank you also for Professor for the nice presentation. I'm Afaf al-Sheikh from Sudan, my roots from Sudan and working in Oman. Actually, we want to know how we can spread the knowledge of sharing genetics and genomic data to be easier for scientists to discover the therapy rather than thinking about the risk of this data, because most of the country in Arab area or in Africa, they didn't start genetic services yet. Some of them, they are thinking about the risk of data, and some of them, they have no services there. We need to know how we can spread the knowledge and let these people in peace and they can start the service in the good way. Thank you.
Well, those are really incredible points. I think this comes to partnership again. So I'm always very happy to tell anyone how we did anything, give you any of our standard operating procedures. I don't regard anything we've learned as proprietary, really. It's about the greater good of getting this established worldwide. At the end of the day, it will come down to a commitment financially to make this work. But I do think partnership can really shorten the lead time to implement that. And that's why I tried to give us a lot of my time to helping others around the world who want to do this, just not to reinvent the wheel. And if we, you know, I've got a whole myriad of things we got wrong. I have no problem with admitting to that or sharing that with you. But I do think, and happy to pick this up offline, that it is important to enfranchise countries that are low middle income to do this, or we will end up with yet another economic divide in the world. And I don't think right now we need that. So I think we ought to redouble our efforts to do that. and perhaps we can pick that up offline.
Thank you so much, Mark. The next question is by Dr. Mariam Boussohail.
Thank you, Professor Mark, for this excellent presentation. Thank you, Dr. Ali. I wanted to ask about your experience with the burden of genome getting a variant of uncertain significance. Yes. How did you deal with it? Like, for example, for us, especially in the field of cardiology, now we started requesting more of genetic studies, and we get all this variant of uncertain significance that keeps more burden sometimes on us and on the family, counseling them and telling them might not, So yeah. With the genome project, did you face, are you facing actually such?
Yes, so absolutely, Mariam. So across 36,000 families, we have about 4,000 variants of uncertain significance. So it's a lot. And this is why I say that the barrier now is not being able to identify variants that may have biological plausibility. It's about being convinced they are biologically real, and about getting us to the level of functional annotation where we can do that. That does require, I think, a global effort. Otherwise, what we'll continue to do with all of our sequencing effort is generate more variants of uncertain significance. And I don't think that's helpful in the clinic. I think that the uncertainty you radiate in your question is very keenly felt. And so how we've handled variants of uncertain significance is to say, we think this could be biologically plausible. We haven't got the evidence. And in some cases, some of the labs that these have gone back to have done their own functional annotation and found they are biologically plausible. And then they close the case and they tell us, yes, this was the variant. Other times it just gets stuck. And that's why I think a systematic effort is needed on this. Otherwise, all of us, our future health, 5 million people, biobank, we're just going to generate millions of uncertain variants. And we won't really be able to resolve them until we get to grips with function. So in my humble view, that's the battleground now. The sequencing is pretty good. We can analyze the genome, but we tend to generate lots of residual uncertainty around the answers that we're producing. So we have to make the step change in that space. So thank you for that question. It's an important one.
Thank you very much for your answer.
Thank you, Mark. I take the liberty of asking the last questions. Actually, I have two questions. One question is about the importance of the information from other analytes, like mRNA, proteomics. Yeah. And if you have planned now, you're planning to sequence more. People are here who are leading large or small sequencing projects. And it might be helpful for them to know what you think about the cost effectiveness of including like other experiments and information from other analytes.
Sally, this is a really good question. So when we started the project, I asked for every rare disease person to have plasma serum and an RNA PAX gene tube, as well as an EDTA tube for whole genome sequencing. So I have 405,000 aliquots on 35,000 people. And during the program, because the program's enrollment was slow and people felt that taking these extra samples slowed it down, that was stopped. We also have several thousand cell-free DNA samples that we collected. So I'm a firm believer that the genome is one part of the picture. I just don't have the evidence for the rest of it. But if you don't collect the samples, you'll never have the evidence. So I would encourage anyone doing this to include multi-omic sampling as business as usual in your program. Otherwise, you're just going to get stuck. And what you'll then have to do is go back to the patient and ask for the samples you could have taken at reduced opportunity cost the first time round. So make your consent broad, make it life course follow-up, be able to recall the patients because there might be a trial opportunity or might need other samples, and be able to let the data be available from a secure data center. In our case, as a reading library, not a lending library. So you can't take the individual data away, but you can work on it in our environment. And that democratizes access. It means you've got a sample repository biobank where you can chip away and get the money because you've got the samples. So you've got a strong argument for doing it. And I think that the future will be multi-omic. And as to cost effectiveness, Ali, well, when we started, people told me from an august sequencing center in the UK that it would cost me £5,670 to do a 30x genome. At which point I could have turned the lights out and gone because the program was completely unaffordable. It turned out that less than a year later, we signed a contract for less than $1,000 per genome. And we've, in the latest contract, which I concluded in 2019, we reduced the genome sequencing price by a further 46%. So there is no doubt that scale can engender price point falls, the surprise more generally. And that does mean coming together in conglomerates of consortia where you can effect a price shift is quite important to the commercial world. The other thing I think is about Ali, because we're still in early days on some of these technologies. I would strongly counsel, people may be wary of this, but I would strongly counsel academic researcher industry partnerships around exploring these technologies. There's huge mutual learning. Our Illumina partnership, where we decided we didn't want to be the sequencer, because I had a room here at my university which had loads of grey boxes in it, and every 3 years they're redundant. So I didn't want that. I didn't want to own a sequencing center. So we rent a sequencing center, which is like the size of a soccer pitch in Kingston, and it's full of Illumina kit. Illumina owns the kit. Illumina runs the facility, and they do a brilliant job of it. And as a result of this partnership, Illumina learned a lot about whole genome sequencing, which they wouldn't have done it if they hadn't worked at scale. And I think it's improved what they've been able to offer to the community. And I think that we should do that in other. I don't think we should work exclusively with Illumina. I think we should work with all the companies. But I also think partnership is a good way forward. And that way, the commercial entity may choose to invest in the program because they can see that they will get knowledge they wouldn't get except by deploying in the old-fashioned way, as some of the providers used to do, the technology, and let you and I try and work out what the problems were and solve them ourselves. I don't think that's a great way of doing things. And I think, partnership is the way forward.
Thank you so much. And the last question, it's a very quick question, but I think important for many people. You saw that we have people from different countries around the world, and some of them are involved with sequencing projects. If you want to list a few important lessons for people who want to start large sequencing projects, or they're doing it, what those will be?
Okay, if you're starting out on this, I would strongly counsel that anyone who is embarking on anything like this talks to lots of people first who know a lot about this. When you do that, A, you learn a lot. B, you also get lots of intelligence about what not to do, and that saves you time. Also, look for partners that are enabling, and you can see there'll be partners. Good partnerships are ones where both sides benefit. Bad partnerships are where one side makes the pip squeak of the other side. It's no longer a partnership and it's a disaster. So I would counsel to gain as much knowledge as you can from the community. This community is pretty good at sharing. I would say it's one of the best scientific communities at sharing. We have a completely open philosophy around data sharing in genomics, and I think that serves us well. I also think you should set your projects up to consider the possibility that you will let others who you don't even know from around the world work on the data, because you will get diagnosis faster for patients. And at the end of the day, you will still manage to publish papers in high impact journals. You just may not publish all the papers. Well, so what? There's plenty of work to go around and plenty of opportunity for others. But if you could be open and allow others in, what you'll get is more diagnosis for your patients, because that's what's happening with our data. And I'm not involved in Genomics England now, so but I am working on the data with my colleagues, like researchers around the world. So I'm now like a researcher around the world. But opening up is very important, I think.
Thank you so much, Sir Mark Caulfield. It was an honor to have you on our Harvard Keynote series on genomic medicine. And thank you very much again for your time and for accepting our invitation. I also want to thank our participants for your time, for input, question, comments, and wanted to remind everyone that our next lectures will be on April 19 by Dr. Huda Zoghbi, and on May 17th by Dr. Tracy Johnson. So please mark it on your calendar, and we are looking forward to seeing you in April and in May for the next lectures. Thank you very much, everyone.